specialized excitatory and conductive system. The cardiac conduction system, CS, also called the electrical conduction system of the heart, transmits the signals generated by the sinoatrial node, the heart's pacemaker, to cause the heart muscle to contract, and pump blood through the body's circulatory system. The pacemaking signal travels through the right atrium to the atrioventricular node, along the bundle of His, and through the bundle branches to Purkinje fibers in the walls of the ventricles. The Purkinje fibers transmit the signals more rapidly to stimulate contraction of the ventricles. The conduction system consists of specialized heart muscle cells, situated within the myocardium. Internodal tracts, heart, anatomy. The interatrial or internodal conduction tracts are bands of specialized myocytes which are believed to lie between the sinoatrial node and the atrioventricular node. Previously, conduction between the two nodes was thought to occur through direct stimulation and conduction between normal atrial myocytes. The latter theory does not explain why the S.A. and impulses arrive at the A.V. and more quickly than would be expected by just atrial myocyte conduction. However, ultrastructural evidence for discrete bands of rapidly conducting myocytes is sparse. Where clear differentiation has been made, the myocytes appear to resemble the Purkinje cells of the ventricles. 3 discrete internodal tracts have been reported. The anterior internodal tract of Bachmann. The middle internodal tract of Wenck-Bach. The posterior internodal tract of Thurell. The figure shows a view of the atria from behind and above. Anterior tract. Leaves anterior surface of S.A.N. Passes posteromedially to interatrial septum and then inferiorly. Splits into 2 bundles. One passes to left atrium, number 1. The other passes posterior to torus aorticus to reach A.V.N., number 2. Middle internodal tracts, number 3. Leaves the posterior and superior surface of S.A.N. Passes inferiorly and medially around orifice of inferior vena cava. Reaches interatrial septum where it passes inferiorly to A.V.N. Posterior internodal tracts, number 4. Leaves the posterior and inferior surface of the S.A.N. Passes inferiorly through the crista terminalis. Passes medially around valve of the inferior vena cava. Reaches posterior surface of A.V.N. Let's repeat it again and remember it. 3 discrete internodal tracts have been reported. The anterior internodal tract of Bachmann. The middle internodal tract of Wenck-Bach. The posterior internodal tract of Thurell. The conduction system of the heart. Stop the slide and give your answers. Check your answers. Organization of the AV node. Organization of the AV node has 3 functional regions. Atrio-nodal, AN, a transitional zone. Nodal, N, the middle part of the AV node. Node His, NH, region, the nodal fibers gradually merge with the bundle of His. AV node. Action potential characteristics of AV node. Long refractory periods, provides protection against high-frequency discharges from atria to ventricles during atrial tachyarrhythmia. Low conduction velocity, physiologic delay of about 0.1 to 0.2 seconds, allows atrial systole to occur before the beginning of ventricular contraction. Accessory conduction pathways. Normally, AV node is the only access pathway for the AP towards the ventricle. When an accessory, aberrant, pathway conducts potential directly from atrium to ventricules, providing a short circuit around the delay in the AV node pre-excitation syndrome. Antigrade conduction occurs over both the accessory pathway and the normal conducting system. The accessory pathway, being faster, depolarizes ventricules earlier. Accessory conduction pathways. The bundle of Kent is an abnormal extra or accessory conduction pathway between the atria and ventricles that is present in a small percentage, between 0.1 and 0.3%, of the general population. This pathway may communicate between the left atrium and the left ventricle, in which case it is termed a type of pre-excitation, or between the right atrium and the right ventricle, in which case it is termed a type B pre-excitation, in old, currently abandoned classification. Problems arise when this pathway creates an electrical circuit that bypasses the AV node. The AV node is capable of slowing the rate of conduction of electrical impulses to the ventricles, whereas the bundle of Kent lacks this capability. When an aberrant electrical connection is made via the bundle of Kent, tachydysrhythmias may therefore result. Accessory conduction pathways. Lone, Ganong, Levine syndrome, LGL proposed pre-excitation syndrome. Accessory pathway composed of James fibers. Characteristic ECG findings of short PR interval, 120 milliseconds, normal P-wave axis, normal/narrow QRS morphology in the presence of paroxysmal tachyarrhythmia. LGL syndrome was originally thought to involve a rapidly conducting accessory pathway, bundle of James, that connects the atria directly to the bundle of His, bypassing the slowly conducting atrioventricular node. However, the majority of those with LGL in whom electrophysiological studies have been performed do not have any evidence of an accessory pathway or structural abnormality. Accessory conduction pathways. Rarer sites include connections between atrial muscle and the conducting tissue within the ventricles, atriofascicular pathways, between the atrioventricular node and the muscle tissue of the ventricle, nodoventricular pathways, and between the conducting tissue of the ventricle and the ventricular muscle, fasciculoventricular pathways. These rarer accessory pathways are sometimes collectively referred to as Maheim pathways or Maheim fibers. The hatched area represents the atrioventricular border. Transmission of the cardiac impulse through the heart, showing the time of appearance, in fractions of a second after initial appearance at the sinoatrial node, in different parts of the heart. Conduction through different myocardial cell types. Sinoatrial node, less than 0.05 meters per second. Atrioventricular node, 0.01 to 0.1 meters per second. Atrial and ventricular conduction pathways, 1 meter per second. His bundle, Purkinje fibers, 2 to 4 meters per second. Atrial and ventricular myocytes, 0.3 to 0.5 meters per second. The Purkinje fibers. Purkinje tissue or subendocardial branches are located in the inner ventricular walls of the heart, just beneath the endocardium in a space called the subendocardium. The Purkinje fibers are specialized conducting fibers composed of electrically excitable cells. They are larger than cardiomyocytes with fewer myofibrils and many mitochondria. They conduct cardiac action potentials more quickly and efficiently than any of the other cells in the heart's electrical conduction system. Purkinje fibers allow the heart's conduction system to create synchronized contractions of its ventricles, and are essential for maintaining a consistent heart rhythm. Any question? Thank you for attention.